Good afternoon, ladies and gentlemen. I'm Professor Ola-Davies, and you're welcome to VBC Module 1 class. Here we are going to go through introductory cell biology and bioenergetics, chemistry and biochemistry of carbohydrates, chemistry and biochemistry of proteins, and cell energetics. Those are the expected outcome. And together with a couple of my colleagues, we'll be taking those lectures over the period of a semester. You can contact me on the above email addresses. Thank you. So what are the expected outcomes? Number one, you have the chemistry and structure of amino acids, peptides and peptide bonds. You are expected to be familiar with the essential and non-essential amino acids and their relevance in health and diseases. Also, protein classification, structure and function, metabolism of amino acid, transamination, oxidative deamination, decarboxylation, and so on and so forth. And lastly, you are expected to be familiar with the urea cycle and its biochemical importance. Stop anyway. So I'll now move to the other slide. There is also the need for us to relate the structure of proteins to their functions, while we also need to understand the importance of amino acid and proteins in biochemical efficiency. Inborn errors of metabolism of some amino acids such as phenylketonuria, tyrosinosis, acaptonuria, albinism, cystinuria also will be taken. And lastly, metabolism of amino sugars. Here are some textbooks that will be available that will be necessary or important for your studies. You have Delvin, you have McKee and James, and you have Straher. This will be important for you to lay your hands on in order to have effective study. So let's quickly start by discussing the biochemistry of proteins and amino acids. Biochemistry of proteins and amino acids. So now we can quickly start by saying proteins are the most abundant organic molecules of the living system, and they occur in every part of the cell and constitute about 50% of the cellular dry weight. Proteins form the fundamental basis of structure and function of life. And the word protein is derived from the Greek word proteus, meaning first place. And this was discovered by the Swedish chemist by the name Berzelius. Proteins are nitrogenous macromolecules composed of many amino acids, and they are very vital in bodybuilding and development. So let's quickly talk about the importance and functions of these proteins. Firstly, proteins are essential constituents of all organisms. They play crucial roles in almost every biological process. They are responsible in one form or the other for a variety of physiological functions, including membrane proteins. We all know that proteins and lipids form the major structural components of cell membrane, and they are the sole source of replacement of nitrogen of the body. Many enzymes and enzyme systems are associated with membrane proteins, that is, components of electron transport system. Also, biochemical catalysts known as enzymes, they are proteins. And maybe I should pause at this point to ask, are all proteins enzymes, or are all enzyme proteins? We'll talk about that as time goes on. Also, we said enzymes, one of the functions of proteins. Enzymes are proteins produced within an organism which are capable of catalyzing specific catalytic reactions. They are biocatalysts which influence the rate of a chemical reaction, usually without undergoing any change themselves. In other words, as they act as catalysts, they do not undergo any change themselves at the end of the reaction. The third function of proteins, as we've been discussing, is hormones. Several hormones are peptides and proteins. Example of that, we know insulin is a peptide hormone. We know that. So, and they also play important role in regulation of metabolic reactions. We know insulin is a peptide hormone, oxytocin is a peptide hormone, calcitonin is a peptide hormone. And we know that these three hormones are active in metabolic reactions. Oxytocin help in, what do we call it now? Oxytocin helps in at the point of parturition. Calcitonin is effective in calcium metabolism. Insulin is a major hormone of carbohydrate metabolism. And we said all these are peptide hormones. They are majorly made up of amino acids. They are majorly made up of amino acids. They are majorly made of amino acids. Some proteins also are known as immunoglobulins, and they serve as first line of defense against bacterial and viral infection. Globulins inhibit proteolytic enzyme and help the immune system of the body. Fibrinogen helps in blood clotting, is actually a blood clotting factor. Lipoproteins are concerned with transport of fat in blood. So that is how lipids are being transported in blood. You don't get to see lipids just that way in blood. They are carried, by proteins. So that shows they actually act in transport system. Hemoglobin is a conjugated protein, that is heme and globin. Conjugated protein consisting of heme and globin. Hemoglobin carries oxygen in the form of oxyhemoglobin in the body. We all know that that is the only way, oxygen is being carried, in the tissues. And then another function of protein is antibodies. We said antibodies. Some antibodies like gramicidin, tyrosidin, and penicillin G are all peptide hormones. Also, we have nucleoproteins. These are conjugated proteins of the cell nuclei. We also have some multiple protein assemblies. These are proteins that contain several compounds which together form a functional unit. Example of that is collagen. We all know what collagen is, which is a fibrous protein found in many connective tissues. Flagella also is one, which are microtubules of flagella made up of proteins. Actin-myosin complex of the muscle also is one, in which you have muscle fibers consist of thin filaments of actin, tropomyosin, and troponin, which make up the light bands, that is the high band, and then the thick filaments of myosin that make up the dark bands in muscle. Another function is under certain conditions, proteins can be catabolized, to supply energy. In the absence of carbohydrates, you see proteins being used up, to provide energy. Those are alternative pathway of energy synthesis when the body is desperately in need of energy. Proteins can be used as a source of energy. Proteins, by means of exerting osmotic pressure, also help in maintenance of electrolytes and water balance in the body. Now, let's quickly talk about amino acid. We said amino acid are the major components of proteins. They are the units that make up proteins. So now talking about amino acid, we said amino acids are a group of organic compounds containing two functional groups, the amino group and the carboxyl group. The amino group, which is the NH2 part of the amino acid, is basic. That is, this is basic in nature, while the carboxyl group, the COOH, is acidic in nature. Amino acids are the building units. They are the building units of protein. We said that earlier on. And proteins are polymers of amino acids linked together by peptide bonds. There are about 300 amino acids in nature, but only 20 of them occur in protein synthesis. And those 20 are actually being called standard amino acids. Only those 20 are actually, very important, and they're the ones that are found in nature. Now, quickly going to structure of amino acid. Don't forget we said basically it's made up of the amino unit, which is the basic unit, and the carboxyl unit, which is the acidic unit. But then we can say that amino acid has 4 different groups attached to alpha carbon. And we know that the alpha carbon is the carbon that is next to the carboxyl group. And these 4 groups are the amino group, the NH2 group, the carboxyl group, the COH group, the hydrogen atom, and the side chain. The side chain is the R, and the side chain could actually be anything. So here you have the structure on the screen. You have the amino group, you have the carboxyl group, which is the acidic side chain. You have the alpha carbon, and then you have on which the R group is being attached, and you have the hydrogen. And this H group, the hydrogen, is common to all alpha amino acids of proteins. You have the amino group, the basic nature, that confers the basic nature on the amino acid. You have the carboxyl group, which is the acidic point. And the side chain is distinctive for each amino acid, whereas the alpha carbon is between the carboxyl and the amino groups. Now, at physiological pH, which is pH of 7.4, the carboxyl group is dissociated, forming a negatively charged carboxylate ion. And amino group is protonated, forming positively charged ion, forming Zwitter ion. In other words, when you have the positive charge and the negative charge at the same point, you have what we call a Zwitter ion. Note that proline is one of the 20 standard amino acids, but proline itself is actually an amino acid and not amino acid based on the structure of that proline, which we're going to look at later. There's also what we call stereoisomerism, which is also, a function that is very important in amino acid metabolism. And this is in D amino acid, you have the amino group is on the right side. We still remember our isomerism. The amino group is on the right hand side, while in L amino acid, it is oriented to the left side, and we can see the basic structure using glyceraldehyde. We have the L-alanine, and we have the D-alanine based on that isomerism. And if a carbon is attached to 4 different groups, it is asymmetric and therefore exhibits isomerism. So if you have any amino acid in which the carbon is not attached to 4 different groups, it is not going to be asymmetric, and then it will not exhibit isomerism. Example of that is glycine, which happens to be the simplest amino acid. The amino acid, possess 4 distinct groups. And like I said except glycine, which is the simplest amino acid. Glycine does not have the R group. It has hydrogen, it has carboxyl, it has amino group. And so it cannot exhibit, it's not asymmetric, and so cannot exhibit isomerism. Thus, all the amino acids, they have optical isomers, and the protein are composed of L-alpha amino acids. I think based on this note, I'll close on this introductory lecture, and then at the next class, we're going to continue with the structure of amino acid. Different structures so as to be able to identify each of them, and then we can go along with the music that I've posted on our VLE platform to identify each amino acid and what they do. Thank you and bless you. Bye for now.
Thymine, tryptophan, phenylalanine, isoleucine, glycine, and there's phenylalanine. These are all nonpolar and proline is as well. There's another that'll add, it's leucine, ain't it swell? Glutamine and threonine and cysteine, tyrosine. These are polar serine too, as well as ferrigine. Now we'll see which ones are charged electrically for you. Glutamic acid is a 1. aspartic acid too. Arginine and histidine are basic